Go ahead and start. So glad to have y'all back. I hope you guys didn't experience any weather-related damage to your home or to your cars. Hopefully your power's back on. As some of you guys probably noticed or saw, I tried to send out messages in our mind letting you know that all the deadlines from last week would be extended. Okay, so any of the due dates or any assignments from last week that you have yet to complete, you have this whole week to get it done. Okay, because I know that not having power or internet for 2 or 3 days could set you back big time, right? And so just get it to me at your earliest convenience. Now, obviously, you don't want to wait too long because then you'll be severely underprepared for our quizzes and our exams. So I'm going to go ahead and share my screen. All right, and so let's take a look at week 12. And I'll need to be making some small changes because of what happened last week, right? And so last Friday, we were supposed to have taken our 6.1 quiz on the structure of DNA and DNA replication. Now, obviously, that didn't happen. And initially, I thought, okay, we'll just try it on Monday. Well, I didn't realize that some people would be without power for so many days, right? I think there are still some families in Burke County that are still without power, right? And might not be getting it for another day or two. So I'm going to extend the quiz. We're going to do that on Friday. So I'm going to combine 6.1 and 6.2 quizzes into just a combined quiz. So the quiz on Friday will cover DNA structure, replication, transcription, translation, as well as mutation. So that will be on Friday. Now, as far as today is concerned, I'm going to kind of take what I would have done with you guys on Thursday and Friday and combine it into a little quick mashup. I'm going to go over some of DNA structure and DNA replication with you guys today. That way you can have an open opportunity to ask any questions. And then we'll take a look at a couple questions afterwards, right? So that way you can see, okay, I get it. Or at least I understand those parts. And there are two videos, right? 6.1 and 6.2. What is DNA and DNA replication? These both can be found in our Unit 6. There are a couple questions with each. They're really short. I think the What is DNA video is like 6 minutes long. Okay, so we're not talking much. Tomorrow and Wednesday are asynchronous. Okay, so tomorrow and Wednesday, they will not be a live Zoom. Tomorrow and Wednesday, your responsibilities are as follows. Make sure to complete those videos because they're due tomorrow. So 6.1 and 6.2, those questions, those are due tomorrow. And then tomorrow while you're at home, let me let this person in. Tomorrow while you're at home, you'll need to watch the lecture videos on transcription and translation. I'll also put a third video on there with some practice problems. So you can practice transcribing and translating DNA into the appropriate amino acids. And so those will be found under the lectures tab. Right now there's 3 DNA, replication, and mutation. So there'll be 3 more posted today that you'll look at tomorrow. And then on Wednesday, there is a interactive that looks at transcription and translation. And I'm going to show you guys that here in just a second because it can be a little buggy, and I'll show you how to deal with the bugs as they arise. That would be something you'd work on Wednesday as well as your study guide. Now, the study guide is not due until, what is it, Friday? Nope, sorry, study guide's due on Monday because that's when our exam will be. But you'll want to have that done for Friday because your quiz on Friday will more or less cover everything that you'll see on the exam. So you'll kind of get like a practice exam on Friday, and then you'll take the real deal on Monday. And as always, there's your little checklist. Okay, so let's take a look at that interactive first. Should be under Unit 6. You can see the first 3 videos there under the lectures. There'll be 3 more, 6.4, 6.5, and 6.6 posted here soon. Here's your study guide, and I even put the pages, or I'm sorry, the questions from the study guide that correlate. And here's the interactive. All right, so go ahead and take that. Oh my goodness, if I can do it. Go ahead and take that link. Then I'll show you what it looks like. And I would pay close attention because I think if this bug happens to you and you watch me do this, you might be thinking, oh, it doesn't work. And you'll send me an email saying it doesn't work. And I'll say, yes, it is. You did pay attention to the video. So when this is what it looks like. You click the little launch button. And it's kind of just like an interactive slide presentation. You kind of just click to the arrows and you can see there's like a little bit of a text box, right? There's some information. There should be some information every single slide. So as you're clicking through, if there's no paragraph or no text, then something's not working properly. And all you need to do is to simply click back to the beginning or simply refresh the page and re-enter or restart the animation. So when you get to like transcription and translation, every slide should have some information. Because I know I looked at it the other day and I saw the pictures and I saw this main text, but these paragraphs weren't there. And if those paragraphs are not there, it's not very helpful. You're just like, okay, I don't know what that is, right? And it goes through step by step by step. And so you should see text on every slide, okay? Now, as I'd said, if that text didn't pop up, simply refresh it or just click back through and start over again, okay? And there's some questions that go with those. I really, really like this interactive, and I really, really, really think it'll help you understand transcription translation. They're very abstract concepts. If you like codes or breaking codes or even translating a foreign language into your own language, you'll find this rather enjoyable. I really do like transcription translation, and I think it's neat that as complex as DNA is, the code is relatively simple, and we've already decoded it, right? So by the end of this unit, you better look at a strand of DNA, and you better tell me what amino acids they code for. And if you know what amino acids they code for, you can inherently figure out what protein that particular portion or gene of DNA codes for. So that's what the interactive looks like. All right, so I'm going to go ahead and quickly go over DNA and RNA. And by no means does this replace the videos I made, because I am literally going to skim the surface really quick, okay? So there's a lot of information in those videos that I posted that I'm not going to address that you do need to know. So DNA is a nucleic acid. It's present in the name. The N and the A is nucleic acid, which tells us that it's made of many nucleotides, okay? And a DNA nucleotide is going to have your sugar, your phosphate group, and your nitrogen base. Now, why is DNA so important? It's kind of like the cookbook or instruction manual for our cells. You're a walking, talking stack of proteins, and each of those proteins is coded for in a gene in your DNA. And it's also a molecule of inheritance or a hereditary molecule. It's like a family heirloom. You got your DNA, 50% from your dad, 50% from your mom. They got it from their parents. And so literally this code is passed down from generation to generation. It's like if your family had a cookbook that was passed down from generation to generation. Now, the DNA does need to be copied, just like, if you were going to make copies of something for each of your family members so they can have it, you'd have to have multiple copies and you want them to be identical. Now, the nucleotides that make up DNA are unique in that the sugar that they contain is known as deoxyribose, and all that means is that oxygen has been removed off of the second carbon here. Now, these numbers right here, you don't necessarily have to have them memorized, but that just gives us a point of reference, especially when we start talking about the directionality, right, or the orientation of DNA molecules. You got 1', 2', 3', 4', and 5'. Now, this 5' carbon right here is what attaches to the phosphate. And if you watch the video, I talk about some neat ways to remember that. Now, there are 4 different nitrogen bases or nitrogenous bases that make up DNA. You got adenine, thymine, guanine, and cytosine. Now, if you look at these nitrogen bases, or let's just look at the nucleotides, you notice that the phosphate and sugars, they're the exact same, okay? There's no variation there. The variation that exists in DNA is actually in the nitrogen bases, right? And you can see that adenine and thymine are not the same, right? You can see that they've got wildly different structures. And we can actually classify them by their nitrogen-based structures. So you've got the purines, that would be adenine and guanine. They have 2 rings, double rings. The pyrimidines, like cytosine and thymine, they're single rings, okay? And then later when we talk about RNA, I'll introduce another pyrimidine known as uracil, and it also has a single ring. Now, the overall structure of DNA is referred to as a double helix. Double is talking about the presence of 2 strands. So DNA is a double-stranded molecule. And the helix is talking about this shape, right? This kind of spiral pattern. So it's like a spiral staircase, if you will. And they're held together by different kinds of bonds. And you can see the nitrogen bases in the middle, and then the outside being the sugar phosphate backbones, right? So if we take the double helix and we kind of flatten it out, you can see this is just like a cartoon rendition. And we can further analyze the molecule. We can look at the individual nucleotides, deoxyribose phosphate, and in this case, cytosine. And you notice that the backbones or the sides are held together by solid lines. These are covalent bonds. These are strong. So the backbone of DNA, the sugar phosphates, are strong, held together by covalent bonds. Now, what holds the two strands together are the pairs of nitrogen bases, and they have hydrogen bonds that hold them together. Now, what do we know about hydrogen bonds? Are they strong? Are they weak? Why do you think in this diagram they're listed as dashed lines? Yeah, they're weak, right? And so the hydrogen bonds can break, and that can allow the DNA strands to come apart and come back together relatively easy. Now, you've also probably noticed that the nitrogen bases don't just randomly pair up. There is somewhat of an order, and we call that the complementary base pairs. You can see that adenine is pairing with thymine. So if you came in one day and I looked at the color of your shirt and I said, you know, that shirt really complements your eyes or really complements your hair color. What I'm saying is they go well together. Adenine and thymine go well together. Cytosine and guanine go well together. And there's a lot of reasons for this. One of which is the number of attachment points. Adenine and thymine have 2 attachment points. They both do. So they like to form 2 hydrogen bonds. Whereas cytosine and guanine have 3. Now, do you think in a hypothetical world that maybe adenine sometimes accidentally pairs with cytosine? Yeah, but is it a really good chemical connection? No, right? And there's another reason why adenine and thymine pair well together, and cytosine and guanine. Adenine is a purine, and thymine is a pyrimidine. And cytosine is a pyrimidine, guanine is a purine. So if you count the number of rings across the center, you got 3 and 3. And that allows DNA to maintain a kind of equal distance between the strands, and some consistency in the size and the shape of the molecule. And if you look here, again, just kind of referring to the directionality, you can see the sugar phosphate backbones, they're kind of going in opposite directions. We call that anti-parallel. The side with the phosphate ending, this would be the 5' end, and then down here, this would be the 3'. And since DNA is anti-parallel, the other side is the opposite. So this would be 3', and that would be 5'. Now, here's when things get kind of wild. DNA replication. What is it? Where does it happen? And when does it happen? Well, when it happens is during interphase. And not just any old part of interphase, it happens during the S phase. So I don't care if we're getting ready for binary fission, mitosis, or meiosis. We have got to make copies of DNA. And that is going to be done with DNA replication. And DNA replication sticks with and follows those complementary base pairing rules. Adenine with thymine, guanine with cytosine. And it uses that to synthesize the new strands, right? Just grabbing these nucleotides floating around the cytoplasm or the nucleoplasm and gluing them together following those pairing rules. And DNA replication is considered semi-conservative. What does semi mean? What is that prefix? Half. And then conservative is talking about being saved. So what this means is that as the result of DNA replication, we end up with 2 molecules of DNA. Now, they are genetically, the code at least, is identical. So these two new molecules of DNA, they have the exact same code, the exact same order of nitrogen bases. If you were to read them, you'd see, okay, wow, it is exactly the same. And not only are they exactly the same as each other, they're exactly the same as the original DNA molecule. But these are replicas. And if I have a World War I replica rifle, what does that mean? Is it the real thing? No, right? Is it modeled after the real thing? Is it probably identical in every single way? Yes. The only difference is one is new and one is the original. And so that's what we see here. As the original DNA molecule peels apart and we synthesize the complementary strand, we end up with 2 DNA molecules that are genetically identical, yet they contain 1 strand from the original and 1 new strand. That's why we call it semi-conservative. Now, there are lots of different enzymes, lots of different proteins that are busy little bumblebees, ensuring that our DNA is unzipped, read, copied, and glued back together efficiently and effectively. So all these enzymes are catalyzing these little tiny reactions. Now, I know this diagram can give anyone an anxiety attack. And I know every time I look at it, I'm like, whoo, let's take it one step at a time. Topoisomerase holds DNA still, right? I mean, so if you get all these enzymes that are trying to read it and trying to build new strands, you need to make sure the DNA is held still or held stable. That's the job of topoisomerase. Keeps DNA from folding back over on itself. Then we need to unzip the DNA, and that's going to be accomplished by helicase. Helicase literally unzips helices. Now, helices being multiple helixes, so it's unzipping, breaking those hydrogen bonds. Now, once we break those hydrogen bonds, we expose the nucleotide, so we expose the code. And the real workhorse here is DNA polymerase. DNA polymerase is going to read the exposed template strands and synthesize new strands using those complementary base pairing rules. Now, how does DNA polymerase know where to start? Before you paint a car, what do you need to do? Put a primer down. And so DNA primase is an enzyme who attaches to the template, the exposed strands, and says, this is where you're going to start. And it lays a RNA primer. It's just a temporary starting point. Then DNA polymerase can attach. It'll start to read the template, synthesizing the new strand. Now, guys, here's the tricky part. DNA polymerase reads DNA from 3' to 5', just in the same way we read a number line. 1, 2, 3, 4, 5. Now, as DNA polymerase reads from 3 to 5, it synthesizes the new strand in the opposite direction, 5 to 3, right? Because again, DNA is antiparallel. Now, why is that important, or why does that even matter? Well, since DNA is antiparallel, that means that nucleotides are in opposite directions. So when DNA polymerase attaches on one side, it's synthesizing a new strand in a continuous form of replication. And this DNA polymerase will eventually catch up with the helicase, and it'll be just riding right behind it. And you get one nice continuous strand of DNA synthesized. Now, on the other side, things get a little weird. Because of the anti-parallel nature of DNA, the DNA polymerase will have to attach. It'll synthesize a new strand, but it'll be moving away from helicase. It'll be moving away from what we call the replication fork. And so it'll have to move this way, detach, kind of sprint back up to the replication fork, reattach, and it makes these new strands in fragments, and they're called Okazaki fragments. And these Okazaki fragments are a form of discontinuous replication. So we refer to that as the lagging strand. It's kind of lagging behind. Now, since we're synthesizing this DNA molecule, or this side of it, in fragments, we need to make sure we have a way of gluing it together. Who is going to glue those Okazaki fragments back together? Which enzyme shows up and makes sure everything's done right? The ligase, right? So the ligase is going to attach those fragments and glue them together, replacing the RNA primer with the appropriate DNA. Now, every time DNA is replicated, we actually lose a little bit of our molecule, right? And so our DNA, or our chromosomes, have these little protective caps. They're called telomere ends. And believe it or not, the telomere ends are just a bunch of junk DNA. But it's important junk, right? It's kind of like the bumper on your car. Does the bumper on your car have any actual function? No. What is the purpose of your bumper? So when you get in an accident, the bumper protects the engine, protects the passenger, protects the important part of your car. Can I go out there and take the bumper off your car and you can drive around just fine? Yeah, you can. There's no problem, right? Yeah, you're right, exactly. If you get in a lot of accidents, it's protective, but it's kind of useless in a sense. So that's kind of how the telomeres are. The code here doesn't code for anything. Look at it. It's just a repeating sequence. In fact, there's a reason why there's lots of G's and C's. They're all triple bound to get, so they like to hold on. But every time our DNA is replicated, a small piece of our strand is lost. And so the telomere end is a protective cap that extends the life of a DNA molecule. I know when I was a kid, I used to always think, why can a turtle, a giant tortoise, live 200 plus years, whereas humans can only live 70 to 80? What determines the lifespan of an organism? What do you think? Why are most of us in this room going to die in our 70s to 80s of old age, whereas a tortoise putzing around on an island chewing on grass gets to live for 2 centuries? It has a lot to do with the telomeres, right? The longer the telomere, the number of divisions possible increases, because eventually, guys, after every time your cells divide, every time your DNA is copied, a little piece of that telomeres end. And eventually, when that useless cap is gone, we start to lose important genes, which means our cells are no longer able to divide. And when your cells are no longer able to divide, you begin to die, right?
Wait, this is going to sound real dumb, but like, can you not, like, are scientists not able to, like, try and, like, separate the telomeres?
Maybe like to put it back on?
Yeah, or like, figure out a way to put the tortoise
telomeres on a human chromosome? Well, so here's another thing. There is an enzyme present in some cells known as telomerase. And telomerase repairs telomere ends. So as every time you make copies of DNA, your telomeres get shorter, telomerase can show up and fix it. There are some cells in our bodies where the telomerase is active, right? Gentlemen, your gonads, believe it or not, your spermatocytes can more or less undergo meiosis infinitely, right? Well, ladies, yours did while you were still in the womb. Ladies, I don't know if you realize this, but you were born with every single egg you'll ever have. You made it before you were even born. That's kind of wild when I first learned about that, right? But how did the cells in the ovaries and the cells in the testes produce so many cells, right? I mean, think about it, ladies. You'll menstruate once a month from as early as 12 or 13 till the time you hit menopause. It's a lot of eggs. Gentlemen, you'll probably produce billions of sperm over the course of your life, right? How is that possible? Endless division. Well, it's because telomerase repairs the telomeres. So that way, when your spermatocytes undergo meiosis and produce sperm, the telomere ends can be repaired. So they can just keep dividing and dividing and dividing. That's why I remember how old James Brown was. You guys know the singer, James Brown? Well, anyways, he had, I think he had a kid when he was like 78 years old, right? Still working. Sperm just as good as it was when he was 15 years old, right? And that's because of the telomerase. Some cancers have active telomerase enzymes. And why is that problematic? Well, if the telomere is kind of like the age limit or division limit, if you can repair it, the cells can theoretically do what? Divide and divide and divide and divide. So what if we could go in and we could turn telomerase off? We could do what to cancer? Stop it right in its tracks. Say, nope, you have an age limit, which would mean that the tumor would get bigger, 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 and it would just stop. Because it would have no way of repairing the telomeres.
So it couldn't do any more damage.
Couldn't do any more damage. Or what if we could go into certain types of cells and turn telomerase on? You could kind of potentially make them what? Immortal. Yeah, right? The cell line, the cells can divide and divide. This could be important. Ladies and gentlemen, if you experience any kind of brain damage, nerve trauma, or spinal cord injury, what is so unfortunate about those 3 events? Yeah, you're shaking your head like it's not good, right? Why? If I cut my arm, it heals, or I break my bone, it heals. But what about your brain? What about your nerves? That's it, man. The number you have in your head is the number you've got. Never anymore. They cannot divide, right? So what if we could trigger division in them, right? Then instead of having irreversible spinal cord damage, we can maybe reverse it somehow, cause those cells to divide. And also you could also add stem cells and give them that job. All right, now let's take a look at some questions. How much of what I just talked about have you retained? There you go. Please join the PD. I had some really interesting discussions with some parents. You know, they're like, I don't understand why my son or daughter's not doing so well. They're like attending all the Zooms. I said, yes, they're signing into the Zoom, but they're not participating. And let me show you. It makes for an interesting discussion when I'm showing a parent like, hey, yeah, they attended that Zoom, but they literally didn't do anything. I asked questions, send private messages to them, no response. They didn't ever sign up for these PDs. So there's 27 of you guys in here. So I should have 27 people join the PD. If for whatever reason you can't because of Wi-Fi issues, just you can play along in the chat. But send me a message right now letting me know. Sounds good. Thank you for letting me know. I'm gonna wait another minute or two. While we're waiting for some people to join, I want to remind each and every single one of you guys, when you're 18, please vote. And I know I've heard students joke like, why don't we learn how to vote in school? Because it's really not that hard. But I'm going to go ahead and just quickly explain it to you because I had a man who was probably in his late 60s, mid 60s, approach me at the polls Friday morning. He came to me and said, I've lived in North Carolina for 45 years. This is my first time voting. He said, I always thought it was going to be more difficult. He's like, I walked in today, I registered to vote, and I voted, and it took less than 10 minutes, right? So when you're 18, you'll probably get a voter registration card in the mail. You literally just fill it out, put it back in the mailbox. A couple weeks later, they'll send you your voter's card. It's got like your name and your address, and they also have a polling location. It's not the only place you can go. It just tells you where is the closest location, right? And so they got locations everywhere. Mine was W.A. Young. It's like maybe 150, 250 yards from my house. But we went to the town hall in Glen Alpine because there you can register and vote in the same day. I was already registered. I had my card, but my wife had just moved here from Tennessee a year and a half ago, and she hadn't registered yet. So I had my card. I walked in. You hand them the card. They check everything. They hand you what literally looks like a Scantron, just like a big piece of paper, right? And you just, there's sections like, you know, presidential candidates and, you know, all the different other running platforms with their names. And all you do is bubble it in, right? And if you don't know, like there was a section on there about Burke County Soil and Water, and you saw those two names and you didn't know either of them, what should you do? Leave it blank. You don't have to, right? Because that's the problem when people are just like, well, I don't know, I'll just pick this person's name, right? That's not how it should be, right? If you don't know anything about those, like, don't, right? And that's also bipartisan. So there's no like political parties associated with that. But the other ones, let's say if you don't know all of the individuals, right? But you are a part of a particular political affiliation, right? You relate more to the certain group or ideals. You can do a straight ballot, which would mean that if you're a Republican or a Libertarian or Democrat, you just go through and bubble all your Democrats or all your Libertarians or all your Republicans, right? The only problem with that is every Republican and just like every Republican, is every Democrat like every Democrat? No, right? Sometimes there are individuals that you might relate more towards, right? And you're like, okay, in this particular situation, you might lean towards this party. But in a different situation, for a different platform, you might be over here. But guys, voting is the easiest thing. Paying your taxes is way harder, right? I mean, downloading some of the apps that you guys have and learning how to use them, I guarantee you, like some of the harder than voting, right? So please go out and vote when you're 18. All right, so I'm my rant. All right, take a look at these questions. You can keep it real short. You can abbreviate if necessary or needed. When you finish, if you would, under the participants tab, just click the green check so I know that you're done. Yes, you can always use abbreviations. Don't worry too much about spelling. It looks like a majority of you guys are close to being done, so I'm going to give it just another minute, and then when you start going over these. These are kinds of questions that you'll see on your quiz. So if you were able to confidently answer these, you're off to a good start. All right. Number one, ladies and gentlemen, how would DNA be classified? What type of biomolecule is it? It's a nucleic acid. So since DNA is a nucleic acid polymer, for number two, what is the subunit or the building block of DNA? What are nucleic acids made out of? Nucleotides. Nucleotides are tied together to form nucleic acids. Now tell me more about nucleotides. What are the three components of a nucleotide? You got sugar, phosphate, and the nitrogen base. Okay, in the case of DNA, DNA's nucleotides contain deoxyribose sugar, phosphate, and then they could have one of the four nitrogen bases, adenine, guanine, thymine, or cytosine, which are what I just labeled, right? A, T, G, and C, adenine, thymine, guanine, and cytosine. What types of bonds hold up the repeating sugar-phosphate backbones? It's rather strong covalent bonds. And then what holds the complementary nitrogenous bases together in the center of the DNA molecule? They're weaker hydrogen bonds. Who likes who more? Who has three hydrogen bonds? Cytosine and guanine, yep. And so adenine will pair with thymine, guanine will always pair with cytosine. I think straight line with straight line, curvy with curvy. To make A and T use straight lines, G and C require curvy lines. And cytosine and guanine have three hydrogen bonds, whereas adenine and thymine only have two. Now, as far as A is concerned, why is it considered semi-conservative? Because when we make those two new molecules of DNA, they are identical to one another and they're identical to the original, but what's different? What's unique about them? Each molecule contains what? Yeah, one of the original and one new strand. All right, so 9. When does DNA replication occur? During interphase. When during interphase? The S phase. Now, in eukaryotic cells, where would DNA replication occur? What are we doing? We're replicating DNA. Where are you going to find DNA in a eukaryote? In the nucleus, okay? So this will happen in the nucleus of eukaryotic cells. All righty, well, if you don't have any other questions or concerns, what you guys need to work on today are 6.1 and 6.2 videos, the video questions, or you can start working on the interactive. Now, if you were out of power or internet all weekend and last week, if you want to go ahead and finish up any of your work from last week and get it turned in, that's perfectly fine by me. If you have any questions, send me an email or remind. Remember, tomorrow and Wednesday are both asynchronous. You've got plenty that you need to get done. And so let's get to it.